So the final set of presentations this morning is going to be a twofer on chronic disease. We have Dr. Gary Wu, who will be talking about inflammatory bowel disease. He's from the University of Pennsylvania. And then we have Steve Clinton from the Ohio State University, who will be talking about cancer. So, Gary, you're going to go first.
Thanks very much. First of all, I'm delighted to be here. I'm learning a tremendous amount, so this has really been fantastic. So what I'd like to do over the next 20 minutes is sort of give you a notion of how we've been thinking about diet and nutrition and using high-dimensional omic technologies. I'm going to use as an example for this our work on inflammatory bowel disease. I'm sorry I forgot my disclosure slide. I will identify the one particular disclosure necessary, but I've also provided it to the National Academies if you want to see it. So I'm a wet bench researcher, so we used different types of animal model systems, and the reason we use that is that there are defined environmental conditions, defined genetics, monotonous diet. And for that reason, there's a high signal to noise ratio, and we're able to demonstrate proof of concept cause and effect relationships with a modest size cohort. By contrast, humans are free living in a highly variable environment, genetic diversity, variable diet. And so one of the big challenges is this intersubject variability that we see in humans. And for that reason, humans are just very noisy. There's a very low signal to noise ratio in a lot of things that we study. Yet we know that small effect sizes over large populations can be highly impactful. I tell people that things that have probably had the biggest impact on saving lives are things like clean water, vaccination. I do believe healthy diet is sort of in that category. So the notion is to embrace the complexity of human biology through the use of high-dimensional analytic technologies together with advanced computational biostatistical platforms. So our notion is that when doing human subject research, and there are a lot of issues and that one has to go through when doing human subject research, is ultimately what we want to do is to match a physiologic response with a lot of different biospecimens that we collect, that we run through very high throughput analytic technologies, and we generate these enormous databases that then we can use our best mathematical approaches, bioinformatics, biostatistics, computational biology, really to understand patterns, identify and characterize biological mechanisms that drive the human response to an intervention. So we have all these different types of technologies. In the field of genomics, when we think about the environment, I particularly focus on microbiome research. We use DNA sequencing technologies. But there are other technologies, immune profiling technologies using mass cytometry, CyTOF being one example of this, which gives you enormous dense information about immunophenotyping in ways that we've never been able to do previously. There are proteomics and obviously metabolomics that I'll touch upon in my talk, looking at many different known molecules as well as many different spectral features. So I'm going to focus on inflammatory bowel disease. Dale talked a lot about this yesterday. It affects a lot of people in the United States, a lot of people worldwide, and the point he made yesterday was our current therapeutic management of inflammatory bowel disease is really focused on the host, and that is deep immunosuppression. So if you were to ask how much of inflammatory bowel disease is due to us, in other words, the genetic contribution to development of Crohn's disease, for example, is at most 30 to 40%, and about 10% for ulcerative colitis, and that's based on a concordance of IBD and identical twins. Now, in an older study a couple of years ago, Judy Cho and colleagues identified 163 IBD-associated genetic loci. If you were to add together the risk imparted by all those loci together, it only accounts for 13% of the disease variance for Crohn's disease and only about 7% of the risk for ulcerative colitis. That means we believe that environmental factors are probably the largest contributor to pathogenesis of inflammatory bowel disease, and we do have evidence for this, that the increasing incidence of inflammatory bowel disease that we see associated with industrialization. This began in Europe and North America many decades ago, and now we're beginning to see this rapid rise associated with industrialization in Asia, in China, in India. Obviously, that's a very powerful environmental effect that is not our genome. And so we think that that's an enormous opportunity. Think about how we might be able to engineer the environment of the gut through diet, through the microbiome, or together to perhaps make our current therapeutic strategies actually work better. So it's important to know that the gut microbiota is deeply dysbiotic in inflammatory bowel disease. It looks a lot different. The reason is that it's a complex microbial community responding to an environmental stress. And inflammatory bowel disease, that environmental stress is inflammation of the intestinal tract. Through nitrate respiration, alternative electron acceptor products, production, oxidative stress, there are many reasons that we believe this to be the case. But I think the fundamental issue is that does this dysbiosis play a role in the chronicity of disease? Well, we do believe that it does, because a lot of these organisms that are overrepresented in a dysbiotic microbiota are disadvantageous to many different types of animal models. And we do have an observation in humans that fecal transplantation, transferring fecal material from a healthy individual with multiple inoculations in a patient with ulcerative colitis can be somewhat beneficial. So with the notion that disease is associated with dysbiosis, and dysbiosis may be playing a role in perpetuating inflammatory bowel disease, the strategy has been to try to change the environment of the gut, to alter that dysbiosis through one of many different methods to try to favor health of the host. And I'm going to focus on dietary intervention. So why do we believe that changing the environment of the gut might be a plausible method to treat IBD? Well, it's because we already have some level of evidence. Dale gave a very nice talk about the evidence for diet and inflammatory bowel disease. We've used for many years metronidazole and ciprofloxacin for the treatment of Crohn's disease, and I gave you that example of fecal microbiota transplantation. I think one of the problems is that these things don't work as robustly and as consistently as we would like them to work. So we think that that's the opportunity to use our best technologies in human subject research to try to answer the following questions. What's the optimal method of administration? Can these modalities be combined with immunosuppression to optimize therapy? What are the optimal clinical settings in which these interventions can be used? And if you do a human subject study, you should collect a lot of biospecimens, use your best analytic technologies to really understand how the human host is actually responding to your intervention. So Dale talked about this defined formula diet. So we utilize this as a therapeutic probe to really understand how diet can be beneficial in a treatment of inflammatory bowel disease. Our paradigm was this. Dale has shown that diet is epidemiologically associated with inflammatory bowel disease. The gut microbiota, for many reasons, unquestionably plays a role in the pathogenesis of IBD. And diet can not only shape the composition of the microbiota, but it leads to the production of many different types of metabolites. So our strategy was to use a diet that we already know works well, try to understand the biological principles by which they work, with the notion that perhaps we'd be able to design better diets for patients with inflammatory bowel disease. So I'm a physician scientist. I'm a practicing gastroenterologist, although I don't do a lot of patient care anymore. But I'm a firm believer that astute clinical observations by physicians in their patients are enormously valuable when thinking about defining and developing hypotheses in the basic science world. And so the clinical observation are that these defined formula diets for the treatment of Crohn's disease are the most effective when they're consumed completely in place of a normal diet. That's why we call it exclusive enteral nutrition. If you start to eat whole food, it doesn't work as well. So implicit in that clinical observation are two fundamental questions that we are trying to answer. Does EEM provide something good for patients with IBD that's not abundant in the regular diet? Or does the consumption of EEM exclude something that is bad for patients with IBD in the regular diet? And I'll give you an example of where we're going with this based on some metabolomic work that we're doing. So when thinking about trying to understand enteronutrition diets and the impact on inflammatory bowel disease, what type of omic technologies would we utilize? Well, if we're going to focus on the microbiome, we can use DNA sequencing technologies, and that's what's in the red box here. Sequencing a lot of DNA tells us about the composition of the microbiota and all the genes that are in those organisms. But another way of thinking about functionality is to look at all the small molecules that are made by these microbes, because maybe they will have an impact on the host because we absorb those small molecules. But perhaps the best way to think about it is an interaction between what is actually there in terms of the microbes as well as the metabolites that they're actually producing. We think that diet is a really good example of this because, as I mentioned, diet not only shapes the composition of the microbiota, but serves as a substrate for these microbes to produce many, many different types of small molecules that we normally cannot produce, that we end up absorbing. After first pass metabolism through the liver, they circulate widely throughout the body. So what I want to tell you about is a study that we called Food and Resulting Microbial Metabolites, called FARMM. The objective was to determine the relationship between dietary composition, gut microbiome composition, the metabolic products that are ultimately present in the gut lumen and in the plasma of humans. So what did we do? So this was an intensive 2-week inpatient controlled feeding experiment where we had vegans, omnivores, and omnivores randomized to consume enteronutrition diet called modulin. Now, I have to say that the omnivores were locked up for 2 weeks in the Penn CTRC because we could not trust them in terms of their diet. But the vegans were actually allowed to be outpatients, and we actually saw that. It was actually something that we saw in our results. So in the first period of the study, we're looking at the impact of these diets on the gut microbiome composition, but then we performed an intervention to study the metabolome. So this was done in healthy human subjects. We've done something like this in the study that Dale mentioned in Crohn's disease called PLEASE, and we think that the intersection between these very two large data sets will give us some idea in the way that diet alters the composition of the gut microbiome, its metabolome and host metabolome that reduces disease in patients with IBD. So this study is part of the Crohn's and Colitis Foundation's Microbiome Initiative. The initiative was to identify new therapeutic targets. Our contribution to this consortium was to use diet as a therapeutic probe to identify those new targets. So this study was funded not only by the Crohn's and Colitis Foundation, but with substantial funding from Nestle as well as Helmsley Charitable Trust. So that's my one relevant disclosure. So let me dig more deeply into the design of this study. So we had 10 individuals in each of 3 dietary arms. And if you look at the interval from here to here, we're looking at diet-induced changes in metabolites and the microbiome. We did this intervention halfway through, where we purged the gut and reduced bacterial load very substantially. And comparing these 2 time points will tell us differences to identify metabolites produced or consumed by microbes. And as the microbiota reconstitutes itself, we can correlate increase or decrease in metabolites with the occurrence of microbes and diet to identify key drivers. So this is an example of something we favor doing in human subject research, and that's a longitudinal prospective study to, again, help us with intersubject variability. We also strongly believe that doing an intervention to look at the dynamic response of diet and the microbiome in human biology gives us a lot more information. So throughout the study, we did shotgun metagenomic sequencing of all the different stool samples. We did fecal, plasma, metabolomics, and we actually did rectal biopsies, isolated lamina-propria mononuclear cells, and have a CyTOP platform to really understand how the mucosal immune system is actually responding at each one of these different intervals here. So I only have time to show you maybe 2 or 3 slides about the results. And this is currently in preparation. So for publication, so because we did shotgun metagenomics, we can annotate the sequence reads to either microbial, which are green, or human, which are blue. So you can see that in the first couple of days of the study, all the reads are actually green because it's all microbial in stool. But halfway through, we flush out the gut, and you see all the microbial reads go away, and it's all blue because that's all human DNA. But then the microbial reads come back. The green actually comes back. Now, we estimate by 16S copy number, PCR, and colony counts that we reduce bacterial load in humans by about 100,000-fold, about 5 logs. So this has never been shown that you could do this before, but it was actually a very meaningful endpoint for us. So the strategy actually worked. What do we actually see in a composition of the microbiome? Well, this is Shannon diversity. This is the numbers of different types of microbes in the gut environment. On a Western diet, it starts high. You flush out the gut. They reduce a lot. And then as the microbiota comes back, it reconstitutes itself. You can see that individuals on a vegan diet, it drops down, but it doesn't drop down as low, and it comes back to a higher level. So that means the vegan microbiome is actually more resilient to this environmental stress than an omnivore's microbiome. On a modulant diet, it drops and it doesn't recover. So what did the composition of the microbiome look like throughout time? So this is a principal coordinate analysis. Each dot is a different stool sample, the entire composition of that microbiome in the sample at that one particular time point. Dots that are closer together are things that are more similar to each other than dots that are further apart, okay? So, ooh, I don't know if I can go back. Oh yeah, here we go. So, I guess I can't go back. Okay, so the day of the study is color-coded on the side here. The symbols represent the different dietary interventions. So the pre-intervention, the dietary phase, everybody is clustered over here. But within 5 days, the people on a modulant diet shift from up here down to here. So remember, all the vegans and omnivores are up here. So modulant has a greater impact on the composition on the human microbiome than the difference between a vegan and an omnivore. So after the intervention, what actually happens? Well, your microbiome now shifts to a completely different state, where now modulant, the people on the modulant diet are completely different from everybody else. So this means that we can use diet to actually shape very meaningfully the composition of the human microbiome. So what about the metabolites? So they're about, this was done by Clary Klisch and Ramnik Xavier at the MIT and Broad. And so there are about 800 small molecules that we know about, and about 50,000 spectral features. So how do we actually think about deconvoluting all that information? Well, the different days of the study are color-coded here in blue. And our biostatisticians came up with this initial algorithm to get us started, and we developed a lot of other tools, but I don't have time to tell you about those. And the idea was, if you looked at the log ratio between one time point and another, if it's greater than 0, that means it's increasing, so we code that as a 1. If it's not statistically different, that's a 2. And if it's actually negative, meaning that the interval is moving down, that's coded as a 3. So at each interval, we could code it with a 4-digit number. So just as an example, PGE2, and this is all looking at feces, the diet doesn't have an effect, so it's coded as a 2. It goes down, and that's coded as a 3 with the purge. It recovers, and that's a 1, and it stays stable afterwards. So the code here is 2312, where you could do it with each diet, hydrocyanamide on a modulant diet. You can see the trends here. The code here would be 2322, so you get the point. So if you look at all the different types of metabolites, the percent proportion of all the different metabolites that can be coded in each different type of diet, and the log abundance is over here and the linear abundance is over here, all the different codes are here. And you notice immediately that on a vegan diet, everybody's clustered in the middle. Well, why is that? Why aren't they at the end? And the reason is that, remember, I told you the vegans were outpatients. We didn't change their diet. So all the vegans had a code of 2 at the very beginning because they didn't change their diets. The other thing was that most of the things did not change, and that's good because you don't want everything to change. The other thing we observed were that there was a substantial number of things on a modulant diet that were coded as a 3. So these things are substantially decreased if you drink a modulant diet. So these may be things that you eat in whole food that may be bad for inflammatory bowel disease. Alternatively, there are other types, a smaller number of metabolites in the stool that actually increase by drinking modulin. Well, maybe these things might be favorable in a setting of inflammatory bowel disease. So in my last slide, I just want to give you an example of how much of an impact the microbiome has on the human plasma metabolome. So this is a study we published a couple of years ago looking at diet and the plasma metabolome and the microbiome of vegans and omnivores. So this is a heat map, and so based on the dietary questionnaire, we look at all these different types of micronutrients, and we can categorize them to certain categories here. And these are in vegans and in omnivores. So vegans are green, omnivores are purple. And things that are positively correlated or increased relative to the other group are red. Things that are decreased are blue. And you can see that based on these micronutrients here, there's a really big distinction. Things that generally are higher in omnivores are lower in vegans and vice versa. So we know that because vegans and omnivores have very different diets. What about the plasma metabolome? So the plasma metabolome, looking at 363 small molecules, is also extremely different, where things that are higher in vegans are lower in omnivores and vice versa. So we can also see this in urine. So all the vegan urine plasma metabolites in composite are over here. All the omnivores are actually over here. So how different are they? So one algorithm is actually to use random forest. It's a machine learning tool that helps identify features that determine a dichotomous outcome. Are you a vegan or are you an omnivore? And by using random forest, we could determine that simply looking at 30 small molecules in blood gives us a 94% predictability to determine whether or not you're a vegan or an omnivore. Now, it's a little bit overkill, but we've used artificial intelligence in this, and we can increase that predictability to about 97, 98%. So how much of this is due to the microbiome? Well, I drew arrows next to the plasma metabolites that are from the microbiome. The point is that the microbiome does have an input, but most of the metabolites are metabolites that are directly an effect on us through diet. So diet has a big effect on our plasma metabolome, and microbiome has an important effect, but it's relatively minor relative to the direct effect on us. So with that, I just want to acknowledge a lot of people that work together with us on this. I've highlighted in red the people that played the fundamental role in our FARMM study. Thank you very much.